Okay, so the electrochemical gradient is established by your sodium potassium pump, right? Um, it uses ATP to pump out 3 sodium and drag in 2 potassium. And because it's using ATP, we know that you're pumping those ions against their concentration gradient. Here's how it works. So ATP binds, the 3 sodiums bind, ATP rips off the phosphate, the sodiums get kicked out, the potassiums get dragged in, and then the phosphate leaves and the potassiums get dragged inside. The electrochemical gradient is established by the pump, but it's also maintained by leak channels, okay? So sodium and potassium have leak channels that they're going to leak down their concentration gradient with, all right? So sodium and potassium move depending on their electrochemical gradient, an amount of leak channels present for each ion. So if you take a look, sodium is highest outside. What's the concentration of sodium outside? It's 150 millimolar. What's the concentration of sodium inside? It is 15 millimolar, right? So, if sodium could go which way is it going to go? Good, it's going to go down its concentration gradient. So it's actually going to go into the cell, right? Oops, I'm trying to get my pen. So it's actually going to go into the cell. And it has leak channels. So sodium has leak channels. Leak channels actually allow sodium to go in any direction, but those leak channels are specific for sodium. And so sodium is going to go down its concentration gradient into the cell. And you also have potassium leak channels. So potassium leaks out of the cell. And so those leak channels are going to be specific for potassium. The trick to understanding action potentials is understanding resting membrane potential. Resting membrane potential is based off of sodium potassium pump and those leak channels. Leak channels are specific for whatever ion they transport. The ion can go in whatever direction. It's always going to move from high to low, right? It's always going to diffuse down its concentration gradient. But if you have a leak channel for sodium, it's not going to let potassium through. If you have a leak channel for potassium, it's not going to let sodium go through. Okay. Take a look at the permeability. So sodiums, how do we know how many leak channels we have? Okay, so the cell has different amounts of leak channels for each ion, and this is going to affect that cell. Potassium is the most leaky because it has the most leak channels. So take a look at those ions. Again, here's the extracellular concentration, here's the intracellular concentration, and here's the relative permeability. Notice that the permeability of potassium, or for that membrane for potassium, is 30 times the permeability is for sodium. So that tells me that those cells have a lot of potassium leak channels and not very many sodium leak channels. Take a look at the permeability for chloride ions in the negative proteins inside the cell. It's almost 0. And so we don't really count the chloride ions, and we don't really count the negative proteins because the permeability is so minute. Okay, so we can just cross those bad boys out. So we can ignore this one, and we can ignore this one. Okay, so which one has the highest permeability? Good, potassium. And so this is telling me that we have a lot of potassium leak channels, right? And so any deviation or any change in potassium outside the cell is going to affect inside the cell greatly. What about sodium? Not very many channels, so it's not going to affect the cell that much, or the extracellular fluid isn't going to affect the cell that much. Okay, so how do we do this? We actually, early, like in the 1960s, they would take squid axons. So the giant squid axons, they have about 2 meter long axons, and you can actually take a sample of the axon and do electrochemical gradient studies on it. And so a lot of the resting membrane potential work was actually done on squid axons. How do we actually figure this out? So notice that it's resting membrane potential. Okay, so this is inside of the cell relative to outside of the cell. So the microelectrode is placed inside the cell, just inside the plasma membrane, and then you have a ground electrode on the outside. And there's a voltmeter attached. This voltmeter is measuring inside compared to outside. Inside of the cell is negative 70 millivolts, and that is relative to outside, okay? And it's because of the potassium. And I'll draw you guys some pictures once we get through this, okay? So resting membrane potential is due to the sodium-potassium pump kicking sodium out and dragging potassium in. So that means that there's more sodium on the outside, more potassium on the inside, and sodium leaking in and potassium leaking out. Resting membrane potential is going to approach potassium's equilibrium. So because we have a lot of potassium link channels, potassium could potentially go to its equilibrium. And if it did, the cell would be at negative 90. But we also have sodium leaking in and we have potassium getting dragged in. So that membrane is not negative 90, it's actually negative 70. So this is how we derive the resting membrane potential. Okay, so let me see if I can draw some pictures for you guys. You're gonna have to, let me see. We'll start with black, okay? So when you're dealing with this, the first thing I want you to do is I want you to draw your cell, okay? So here's your cell, right? It's gonna be really crude, I apologize. And you want to draw your sodium potassium pump. So here's my sodium potassium pump, right? And pick different colors for sodium. Let's see, and for sodium and for potassium. Let's see. So we'll do blue for sodium. Okay. So how many sodium are we kicking out? Good. 3 sodium out. And so I'm going to put those right here. And then how many potassium are we dragging in? Good. We're dragging in 2 potassium. Okay, so we're going to put the 2 potassium right there. Okay, what do we need to drive this pump? Okay, good. We need ATP. So pen color. So draw your picture. And again, you need to be drawing this and practicing this so that you understand how this occurs. So here's our ATP pump. This needs ATP to kick out 3 sodium and drag in 2 potassium. So you're kicking out sodium where it's highest, right? So where is sodium highest? Sodium is highest outside of the cell, right? So let's go and put that there. So we have high sodium. Where is sodium lowest? Good, so sodium is lowest in, oh, whoops. Sodium is lowest inside the cell. Let's try to do that again. Okay. And then what about potassium? Where is potassium highest? Good, so we're dragging potassium in. We're using ATP to do that. So potassium is highest inside the cell and it's lowest outside the cell. Okay, go back to the picture that you drew before. Does that align with that picture? So sodium's concentration outside is 150. Sodium's concentration inside is 15. Potassium's concentration is 150 inside and then 5 outside. All right, so the sodium potassium pump causes sodium to get kicked out and potassium to get dragged in. Plus, it's also going to kick out 3 sodiums, which is 3 positive charges on the outside, and it's going to drag in 2 positive charges on the inside, okay? So 3 positive charges outside, 2 positive charges inside. Let me ask you a question. Which side has more positive? The outside or inside? Good, the outside of the cell membrane. Which side has less positives? The inside of the membrane. Okay, which side has more positives? The outside of the membrane. Which side is relatively negative? Because it has less positives. Good. Okay, so the inside of your cell is relatively negative, and we're just going to put a negative down here, because of that unequal dragging of those ions. So you kick out 3 sodium and you drag in 2 potassium. Because you do that, the inside is relatively negative compared to the outside. Now, how do we get the negative 70? Okay, so how do we get that negative 70? Whoops. See if I can undo that. Oh. Guess not. Okay, I'll draw it again real quick. All right. So we get that negative 70 from potassium's equilibrium potential. So let me draw this again. So here is our cell, right? Here's our sodium potassium pump. We're going to kick out 3 sodium, right? So, and we're going to drag in 2 potassium. So, and we need ATP to do this. So because we need ATP to do this, we know that we're pushing sodium where it's highest. So there's high sodium here, right? And we're dragging potassium in, so we know that potassium is highest here. Whoa, let's see. Let's try that again. Okay, so let's go back to the 2 potassium. Okay, so we're dragging potassium in, so potassium is higher here, right? And sodium's obviously lower here. And then potassium is lower on the outside. Okay, so this pump sets up separation of charge. How does it do it? It makes sure that sodium is highest here and potassium is highest here. It also makes sure that we're kicking out 3 positive charges on the outside and that there's 2 positive charges getting dragged in, right? Because we're pumping out 3 sodiums and we're dragging in 2 potassiums. So because there's unequal distribution of positive charges, the inside is relatively negative compared to the outside, okay? How do we get the negative 70? If we looked at potassium, let me erase this. If we looked at potassium and we let potassium go till its equilibrium, it would leak out, right? So this cell, remember the permeability of potassium? So I'm going to draw the potassium channels, right? These are all leak channels, non-voltage gated channels. These are leak channels. So which way is potassium going to go if it could? It's going to go down its concentration gradient, right? So potassium is going to go down its concentration gradient. If we let potassium go to equilibrium, if we ignored everything else and just let potassium go to its equilibrium, inside the cell would be at -90, okay, millivolts. But that's not the case. Why is it not -90? Because we have potassium getting dragged in by the pump, and we have sodium going in via sodium leak channels, okay? There's not very many sodium leak channels, but a positive ion coming in, that's gonna make this more positive. And the potassium coming in is gonna make this more positive as well. So this, instead of -90, it becomes what, guys? Okay, it becomes negative what? What's resting membrane potential? Good, -70, okay? So this is -70. So let's recap. Your sodium potassium pump has to have ATP to work. So you have to be living, right? That's going to pump out 3 sodium, drag in 2 potassium. That's going to set up the separation of charge. So you're pumping sodium where it's highest. You're pumping potassium where it's highest. Okay. And because you're pumping 3 positive ions out and you're dragging through 2 positive ions in, this is negative compared to the inside of the membrane is negative compared to the outside. And because we have a lot of leak channels for potassium, potassium is rushing out. A positive ion rushing out is going to make this -90, right? But it's not at -90 because we have sodium leaking in and we have potassium getting dragged in. Okay, so this is how we establish resting membrane potential. Okay, if we had to plot it, it would be at -70 for nerve and skeletal muscle. I want you to draw this out a couple of times so that you understand it. It's important to understand resting membrane potential because you will see times when, let's see, you will see times when your patient has a lower potassium than normal or a higher potassium than normal. So if it's really low, then potassium it is going to rush out faster, and this will be more negative, okay? Making it harder to excite. If the person has high serum potassium, right, then this is going to stay inside longer and this is going to be more positive, okay, so easier to excite. So low potassium, right? So low potassium outside, potassium is going to go out faster, and that's going to make it more negative. So inside, so is it going to be harder or easier to excite? Good. You will see this with coordination and cognitive ability. So it'll be harder to pick up stuff. It'll be harder for that person to think through stuff, more mental confusion. What if they had a higher potassium outside? So let's say they were taking some diuretics kick out potassium as well to lower blood pressure, and some diuretics increase potassium. So what if, or some meds increase potassium, what if you have a lot of potassium extracellularly? That's going to prevent the potassium from leaving, right? So more potassium here, this potassium isn't going to want to leave. And so the inside is more positive, right? And so the closer you get to -55, the easier it is to excite. Okay. And so that's why you have to be really careful about potassium extracellularly. So potassium normal range for plasma is usually 4.5 to 5.5 millimolar, right? Okay. So let's move on. So that's resting membrane potential. Let's get into excitable cells. So resting membrane potential is the most important. You have to have resting membrane potential before you can have depolarization, repolarization, and hyperpolarization. Okay, so the 4 phases for excitable cell would be resting membrane potential. These are for nerves and muscles. Then depolarization to threshold and depolarization producing an action potential. And then repolarization and hyperpolarization. Okay, so let's go over that. Here's your time in milliseconds. Here's your membrane potential in millivolts, right? This is negative 70. So this is analogous. This nerve is in resting membrane potential. I like to think of this as on standby. So you turn your computer on, your computer's in sleep mode, it's still on, but it's just waiting for you to move the mouse or you know, hit the keyboard. And so this is the same thing as a nerve. So your nerve is sitting there at negative 70 and nothing happens until it gets zapped by another nerve or until you think about like moving a skeletal muscle or something. And then that stimulus is going to open sodium voltage gated channels. Okay, so let me draw on this. Pick a color that you guys can see. Let's do blue. Blue seems to work really well. Okay, so where's my pen? So the stimulus, you're actually going to open some of those sodium voltage gated channels. Okay, I'm going to put V, G, C, open. Some. Okay, so VGC stands for voltage gated channels. So some of those sodium voltage gated channels are going to pop open. So look at the picture that you drew and draw some sodium voltage gated channels. And sodium is going to rush where? It's higher outside and lower inside, so it's going to rush into the cell, right? So sodium is going to rush into the cell. And when sodium rushes into the cell, the inside is going to get more positive. Okay, if you have enough sodium rushing inside the cell that the cell goes from negative 70 to negative 55, then you hit threshold and you're going to produce an action potential. All right, negative 55 is sort of like the magic voltage. Okay, if you hit negative 55, then all of the sodium voltage gated channels pop open in that region and sodium just floods into that cell and depolarizes it. Okay, so this is called depolarization. Sodium is going into the cell using what channels? And be careful, we have 2 channels. We have leak channels and we have voltage gated channels. Good, so it's going to be your voltage gated channels. It's not that the leak channels aren't there, they are. The pump and the leak channels are there. They're still doing their thing. They're always doing their thing. Remember, your computer is on, whether you're surfing the internet or typing up a paper. So this is just, this overshadows the resting membrane potential because it's such a strong change in action potential, okay? So you get some type of a stimulus, some of those sodium voltage gated channels open, sodium starts rushing in. If you get enough sodium inside the cell that the inside hits negative 55, then all of the sodium voltage gated channels in that area pop open and sodium rushes into the cell. Okay. Students have a hard time understanding threshold. So let me explain this to you. Let's say I have a bowl of macaroni and cheese and I put some salt on it, right? And I mix it up and I taste the macaroni and cheese. That sodium is actually going into my taste bud cells and it's depolarizing my taste bud cells, or you're getting an accumulation of a positive charge inside the taste bud cells. Okay, if you get enough positive charge that it goes from negative 70 up to negative 55, you're going to hit threshold and you're going to taste the salt. If you don't get enough accumulation of positive charge and it doesn't hit negative 55, you're not going to taste salt. Okay. So I put some table salt on my macaroni and cheese and taste it. And I'm like, oh, I can't taste salt. Did my taste bud cells hit negative 55? No. Okay. So then I add some more salt onto my macaroni and cheese, mix it up and taste it. I still don't taste salt. Did my taste bud cells hit threshold? No, they didn't. There wasn't enough sodium accumulating in that taste bud cell to hit negative 55. Okay, so then I take the whole salt shaker and dump it in the macaroni and cheese, mix it up and taste it. Oh, okay, now I taste salt. Did my taste bud cells hit threshold? They did, right? So there was enough sodium ions inside the cell that the charge went from negative 70 to negative 55. It became more positive. Once you hit negative 55, all those sodium voltage gated channels pop open and sodium rushes in, okay? And that causes depolarization to the peak of the action potential. This whole thing, this upward swing and downward swing, is called an action potential. Okay, so what happens at the peak? If you guys remember back to when we talked about sodium voltage gated channels, the sodium voltage gated channels, become inactive, okay? So they are weird. They are the ones that have different settings. So sodium voltage gated channels open, right? So these guys open and then they become inactive. And they are actually inactive this whole time period. How do we know that they're inactive, or what is the name that we give this? We call this absolute refractory period. Okay, let me draw my X's and I'll show you where absolute is. Okay. It's right here. So absolute refractory period. So the whole time these guys are inactive, this nerve is in absolute refractory period. Okay, what does that mean? If these guys are stimulated again, they're not going to pop open because the voltage gated channels are inactive. And if they don't pop open, then they're not going to produce another action potential. Right? Let's take a look at what potassium does. So let me change the color. Let's try this one. Okay, so at the peak, then what happens? So potassium, no, sorry guys. Adventures in labeling your PowerPoints and drawing pictures with Professor Merlot. Yay. We'll do black, okay? So at the peak, potassium voltage gated channels open. Okay, and so potassium voltage gated channels open, and potassium is going to go where? Look at your picture. Where is potassium highest? Potassium is highest on the inside, and it's going to leave. So potassium goes out, right? So potassium is rushing out. A positive ion rushing out, what is that going to do to the inside of the membrane? It's going to make it more negative, right? So that positive ion rushes out and that membrane gets more and more negative. It's actually going to hit around negative 88, okay? It gets dangerously close to potassium's equilibrium potential. And if it hits potassium's equilibrium potential at negative 90, this cell is in essence dead because it's at equilibrium. It's not going to do any work, right? Okay, so potassium leaves using the voltage gated channels. Potassium rushes out, and then eventually, right around in here, potassium voltage gated channels close, right? Okay, they don't have an inactivation phase like sodium does. And so how does a cell then get back to resting membrane potential? Good, it's gonna be your sodium potassium pump and your potassium leak channels, right? and your sodium leak channels. Okay, let's go back to sodium. So if you notice the sodium voltage gated channels are inactive for a certain amount of time, this makes sure that you can't get another action potential. Okay, relative refractory period is up here. So relative refractory period is when those sodium voltage gated channels have changed to uh, closed. So you can stimulate them to pop open again. Let me see. Okay, so relative refractory period means that some of those sodium voltage gated channels that were inactive are reset to closed. Okay, and so if enough of them are reset to closed, when you zap it, you're going to get another action potential. Okay, so let me recap. And I want you guys to draw this out and label them as well, right? Let's see, laser pointer. So resting membrane potential, sodium potassium pump, sodium leak channels, potassium leak channels. This gets some type of a stimulus, and some sodium voltage gated channels are going to pop open and some sodium rushes in. If we have enough sodium rushing into that cell that it hits negative 55, then you're going to generate an action potential. Okay, that's threshold. And so what ends up happening once you hit threshold is that sodium voltage gated channels pop open in that area of the axon. Sodium rushes in, and you get this upward swing of the graph, right? So this is called depolarization. Sodium is going into the cell using the voltage gated channels. And then at the peak, sodium voltage gated channels become inactive. Potassium voltage gated channels pop open and potassium rushes out, causing the downward phase. Once it goes below resting, we call that hyperpolarization. Okay, so below is hyperpolarization. And then at the bottom, potassium voltage gated channels close. What brings us back to resting? Your sodium potassium pump, your sodium leak channels, and your potassium leak channels. We have more potassium leak channels than sodium, so it's primarily due to the pump and potassium leak channels. How do we make sure that that action potential goes in one direction down the nerve, from, let's say, the nerve head to the tail, if so to speak, right? Or the cell body to the axon terminal region, right? It's because of these sodium voltage gated channels being inactive. So when this nerve is in absolute refractory period, that means that the sodium voltage gated channels are inactive and they're not going to pop open. And so you're not going to generate another action potential. Once we get into relative refractory period, some of them start transitioning to closed. And once enough of them are transitioned to closed, that you zap it and they pop open and they can generate another action potential. Okay. The purpose of absolute refractory period is to make sure that that action potential goes one direction down the nerve. Okay. Okay. You can read this and test yourself. Here's the sodium voltage gated channels. So at negative 70, they are closed. Then they pop open, just a little bit more positive than that. Sodium rushes in, and then they become inactive for a certain amount of time. And then after a certain amount of time, they reset back to closed. Then they can get popped open again. Then they go inactive, and then they reset to closed. Okay. So depolarization is to make the cell more positive, the inside of the cell more positive, right? And so you can say less negative, that's fine, more positive, same thing. And so sodium is going to rush in, causing that depolarization. Okay. So once depolarization has started, it can die down, or if it hits negative 55 inside the cell, then an action potential will happen. Okay. If it doesn't hit threshold, no action potential will happen. So you can take a look at that. Action potentials operate on an all or none principle. So the stimulus has to open up enough sodium voltage gated channels so that the inside of the cell hits threshold, which is negative 55 for your nerves and skeletal muscle. If threshold is reached, you'll get an action potential. If threshold isn't reached, you won't get an action potential. Okay, this is just like the macaroni. So if I put salt on my macaroni and I don't taste salt, I didn't hit threshold, so I didn't get an action potential generated to the brain. If I put enough salt on my macaroni and I hit threshold with my taste bud cells, then I'm going to generate an action potential and my brain's going to interpret that as salt. Okay. Once an action potential has been generated, the amplitude of that wave will be the same for all action potentials generated. So how do we get stronger stimuli? We send more action potentials down the line. So the action potential waveforms aren't any bigger. It's that we send many action potentials to get the brain to understand that it's a stronger stimulus. Okay. At the end of depolarization, the start of repolarization, your sodium voltage gated channels become inactive, so no sodium is going in, and potassium voltage gated channels open. So now it's potassium's turn. So potassium is going to rush where? In or out? Good, it's going to rush out. Okay, so right here, potassium is going to rush out. So we're in repolarization. It makes inside more negative, right? Potassium voltage gated channels open. They're going to stay open for a certain amount of time and potassium rushes out. Then we're at hyperpolarization. So what happens at hyperpolarization? It's to make inside excessively negative. Potassium voltage gated channels close during hyperpolarization. Potassium is still rushing out and the membrane potential dips down past resting. Okay, so ultimately, all of potassium voltage gated channels are going to close, preventing potassium from hitting its equilibrium potential, right? And then what brings it back? Your sodium potassium pump plus your link channels, okay? And so this is hyperpolarization. You can skip this. This is just showing that depolarization is dependent on sodium and that repolarization and resting is dependent on potassium. And so those are the those channels in sodium and potassium, those phases of the action potential are dependent on those ions. Okay, so take a look. So if we're looking at how an action potential is generated, let's say action potential is generated at the axon hillock, that depolarizes first, and then this region right next to it depolarizes, and then this region right next to it depolarizes, and then this region right next to it depolarizes. How do we make sure that the action potential doesn't go backwards? Okay, so take a look at the bottom of this. Absolute refractory period prevents another action potential. So the fact that these guys just depolarized and now they're in absolute refractory period means they're inactive. They can't depolarize again. So that action potential has to jump to this region. This region depolarizes and then it's done. It can't be stimulated again for a certain amount of time. Then this region depolarizes. And so you're making sure that that action potential goes down the nerve the way that it's supposed to. Okay, you can take a look at the same thing. Okay, there are two types of refractory period in excitable cells. Absolute refractory period, where sodium voltage gated channels are inactive, no matter how much voltage you zap them with. And make sure that that action potential goes down the nerve axon the way you want it to go. And then relative refractory period, some sodium voltage gated channels are closed, some are still inactive. You can zap it with a stronger stimulus or stronger than threshold stimulus, and that you will get another action potential. Okay, so here's absolute refractory period, here's relative refractory period. Take a look at this picture. So here's the first stimulus, you get an action potential, right? If you stick the second stimulus, even though it's stronger, while the cell is in absolute refractory period, you're not going to get another action potential. If you do a stronger stimulus after, when the cell is in relative, you will produce an action potential. It's not going to be as strong of an action potential, but you'll still get one. If you wait until that nerve is fully recovered, then you'll get another action potential. Okay, so I want you to figure out how cells or how action potentials are affected by high or low potassium outside the cell and high or low sodium outside the cell. Okay. I want you to draw your pictures and I want you to go through this. You can also do on Mastering AMP, there is a dry lab that the physiology students did. If you haven't done this, you can do resting membrane potential. So it's www.masteringamp.com, and then you can log into that. And let me see, do black. So it's www.masteringA&P.com. Okay, and there's a neurophysiology dry lab. I think it's experiment 3. And that helps you do a virtual lab of a squid axon with resting membrane potential and changing the different ion concentrations. Okay, thank you, and I will see you soon.